So now that you have an idea of what all the parts of the microscope do and where they are, let's talk a little bit about what you're going to be doing in lab. So when you are practicing with the microscope in lab, you are going to be playing with and noticing the different types of magnifications, what you can see, what you can't see, what happens to light, what happens to magnification, right? So I'm going to run through all of that a little bit here. So the first thing to review from the last video is that in our microscopes, we have 3 objective lenses, right? So there's the scanning power at 4x, low power at 10x, and then 40x, which is considered high power. Now we also have those ocular lenses that we talked about, right? So again, our microscopes have 2. They are binoculars, if you will, that are also at 10x, which means if we are looking at something through the oculars and the objectives, we're not just looking at something at 4x, right? We're looking at it at 4x and 10x. So the way that we can figure out what's called total magnification, how much magnification are we actually using if we are using the objective lenses and the ocular lenses? So it's simple, really, like we're just magnifying them together. So we have to multiply the 2 powers together, right? So we're going to take the ocular power of 10x and we're going to multiply that times the objective power. So if you're on scanning at 4x, the ocular is going to be at 10x, right? So it would be a total magnification of 40, right? 10 times 4. So if we use the example of ocular lenses being 10x and the objective power being 100x, in order to figure out our total magnification, all we have to do is multiply the 10x of our oculars times the 100x of our objective, which means we are looking at something at 1,000 times what we would see with the naked eye, right? So in our microscopes, we have a 4x times 10 at 40x, a 10x times 10 at 100x, and then a 40x times 10 at 400x. So you should be able to calculate this if we give you an example, right, of the ocular lenses and the objective lenses. So it's something to practice in your math. So one of the things we also see happen as we use and increase our magnification. So as we go from 4x to 10x and 10x to 40x, the magnification does increase, right? So we're seeing things at a higher magnification, right? Smaller objects are appearing larger. Our resolution also gets better, which means we can see those smaller objects a lot clearer. So that's one of the correlations that you should see as you go through this lab. So the other concept that's in here, and this picture should look familiar as it's straight from your lab book, is about field of view, or what's called the FOV. So the field of view is basically just how much space are we able to see when we are looking through the microscope. So in our microscopes, the field of views are round, right? In most microscopes, the field of view is round. So as we look at it, we can actually estimate about how large the field of view is, and that will help us to determine how big the stuff inside of the field of view is. So this image shows you kind of what happens as we zoom in, right? So this giant gray circle around here is all of the area that you would see if you were looking through the scanning power, the 4x. Then as we zoom in, we lose some of that. So you see how much of the area we actually lose as we zoom into 10x, and then how much more we lose as we zoom into 40x. So as we zoom in, using our stronger powers, we lose the amount of space we have, which means the field of view decreases. So as the magnification is increased, our field of view decreases. And remember, we're basically doing this so that a resolution increases, so we can see those smaller things better, but we do lose space, right? We don't see as much of the area as we would at a lower power. So I often compare the microscope to a camera because I think more people have used the camera on their phone than they have a microscope. So when we zoom in on our camera, right, when you think about zooming in to take a picture, you are losing all of the space around something, right? It's one of the reasons we zoom in, right? If you want to take a picture of just your cat, you don't want the area around the cat. So the field of view is going to get smaller. Now, the other thing that we can apply that to is the second concept that's discussed in your lab book called depth of field, or the DOF. So the depth of field is basically how much of the object is in focus at one time. So not necessarily how much of the object you can see or not how much of the area you can see, but just the specific object. So again, as we zoom in with our microscope and magnification, the depth of field goes down. Basically, you can't see the whole object clearly when you move from 4x to 10x or 10x to 40x. So again, I think about this in terms of a camera, and I took this picture from Wikimedia, kind of shows you what happens when you're using a camera, right? So if you're taking a full picture of the entire scene, right, as you look through your camera, you can see everything there. In this case, the dog, the person, and the tree. And then if you want to take a more specific photo, you focus specifically on one object. In this case, the person. So you are taking a picture of the person, then the dog gets a little blurry, the tree gets a little blurry, because you are only looking at the person, which means the depth of field has decreased as you zoomed in, as we used a higher power. Same thing's true in our microscope, right? So when you zoom in on your microscope, you might be able to see that there's a lot of stuff on your slide, but you're only going to be able to really focus on one thing as we zoom in, and hopefully that is the one thing that you are aiming to actually focus on. So in your lab, you are going to be given your own microscope. You are going to be given slides in order to practice on. Now, the classic example of the practice slide is a glass slide with a typewritten E on it, right? So we literally print out an E, we cut it out, and somebody has to put it onto the slide. That is not you, right? These are prepared slides. So these prepared slides are what you're going to practice with first. And you're going to go through the steps, and your instructor is going to walk you through how to actually focus the microscope so you can practice this. So as you do this, you are going to be paying attention to what happens to how the light changes when you focus, how when you move the stage and the stage clips and the slide, how does that affect the movement of the actual E? So what you are going to learn here, so when you see the E, so this is what the E would look like when you're just looking at the slide without the microscope, right? So this is what you're going to slide onto the stage. You will have an E. Then you are going to get it into focus under the microscope. So when we focus microscope, as I'll talk about in a second, we always start at the scanning power. We start at 4x. It makes things easier. So when you start at 4x underneath the microscope, what you will see in your field of view is the E. Now, the E does not look the same, right? What you notice here is the E is not just upside down, right? So obviously it's upside down, but it's also backwards. This is what we call inverted, right? So the lenses that are in the microscope, the way light passes through them, it actually inverts the object. The same thing happens in our eyeballs, actually. Like when light passes through our eyes, we get everything inverted as well. Then our brain fixes it. So with the microscope, there's no brain to fix it. We have to fix it, right? So we are going to see this E as upside down and backwards, or inverted, which means when you move the stage to the right, it's going to be backwards, which means when you're looking at it, it will actually move to the left. If it's going to move to the left, right, the image will actually move to the right. So everything is inverted underneath the microscope, and it does take a little bit of practice to get used to that. So then what you're going to do is you're going to zoom in on your E, right? So you're going to focus on 4x, and then you're going to move it to a higher power. And then you will see, the E. You'll see what kind of looks like E, part of the E, but not really the E, right? We lose the whole image, right? So as you zoom in, again, resolution will get better. You will see a lot more detail of the typewritten E, but you will not see the whole image, right? And that's part of what happens as you are practicing learning how to use the scopes. So when we focus our microscope, you're going to take that E slide first. You're going to slide it onto the stage. You're going to put it over the light by using those mechanical stage controls that I talked about in the first one, right? The little stick that hangs off the side of the stage on the right-hand side of our microscope. You're going to move the slide around, all right? And you're going to get whatever you want to look at, like the E, right in the center of the light. Then you are going to make sure that your 4x, remember that's the scanning power, is aimed down, and you are going to then use the coarse focus knob in order to raise the stage basically all the way up. Do this gently, but you're going to pull it basically all the way up, right? That's how our scopes are going to focus. And then once you get up there, you're going to slowly lower the stage until you can actually see your specimen, the E. And then you're going to use the fine focus. Remember that knob was like kind of on the outside of the coarse focus, to adjust this very, very slowly and make it so you can see your image well. Now, our microscopes are pretty cool in the fact that they're what we call parfocaled and parcentered, which means that the objectives are basically all proportional to one another. So when you are focused on 4x, you don't really have to touch anything and you can go right up to 10x. So you'll just move the 4x objective out of the way and the 10x will slide in, and you will then be able to kind of see your object. It's not going to be clear, right? I mean, you'll be able to see it. And then because of that, you don't need to touch your coarse focus like ever again. Right? Your coarse focus is not needed after that point. You will only need to use that fine focus knob. That will allow for you to have very nice control to make it picture perfect at 10x. Then, once you are going to get that in focus, you want to center the object because it's not always perfect. So once it is centered, and once it is focused, aka par focal, then par centered, then you will be able to slide it right into 40x. Now with our microscopes, it's very proportional, and that also means that the objectives will not scratch the stage and the slide that you're looking at, which is different than some other microscopes. So be aware of that. So then once you're on 40x, you are going to use, again, only the fine focus. If you touch the coarse objective knob, it is not going to end well. You will lose your object very quickly, and then it'll be really hard to come back at that. So once you get it on focus in 40x, you will notice that your light has decreased. So as we zoom in, our light levels decrease. So you'll have to adjust that to your liking. Using the condenser diaphragm is what I would suggest, but you might need to play with the rheostat as well, just in case. So you will see as you zoom in that resolution increases, field of view decreases, depth of field decreases, and light levels will decrease. So that will give you a clearer picture of whatever you're looking at, but you are kind of losing some things as you zoom in as well, right? You lose the big picture and you use light, which is why we have the adjustments the way we do on our light levels.